told you last time that you're going to have one lecture on insects and only one. And we used to hand out this nice bulletin, which is one of the best the university ever put out, with all sorts of color photographs and what have you in it, about the insects and their history and their life and all this business. But because of this stupidity about DDT being included as treatments throughout here, the university ordered them all to be destroyed. So you can't have that anymore. But we do have on reserve and 6 copies in the main library and 3 copies in our Vit library for 2-hour reserve, a set of 6 pages, photos, color photos of the most important insects. And inside that is stapled, inside the folder that has the pictures, is stapled a copy of the 1973 control measures recommended. So what you're going to be held responsible for recognizing those 6 insects, and I put insects in quotation marks because red spiders are there, and as you know, spiders are not insects. That's something that a lot of people, including myself, wonder about. But there's red spider, 2 types, and 4 of the main problem insects included in that. So you will be held responsible for knowing those, recognizing them, and giving at least 1 treatment. Only 1 treatment is all you need. And
you might mention that they could purchase the series of 6 if they write down the Berkeley.
Yeah.
I think it's $600.
That was the next statement. Thank you, but I almost forgot it. We checked it out here to see if you could get these sets here at Davis, and I don't know why you can't buy them at Davis. But these are classic color photographs of these insects. I bought 3 sets for myself because they're going to go out of print as fast as right away, I think. Rather than each one of you having to write to Berkeley, if some of you want them, maybe if you just go into the office to Judy, that first girl you meet when you walk in the office, and give her your name and $1, I'll order them all at once. So just ask for Judy in the office, give her your $1 and your name, and then I'll just order them all and then get them for you. Oh, they come up within 3 or 4 days. Usually the bus goes down. If they're really fishing, they come back the next day, but let's say 3 or 4 days. So, but that won't give you the treatment for it. And they have about 20 or 30 of those 1973 disease and insect controls up in the extension office, but they did not have enough to give everyone in the class one. So rather than have some with them and some without, I said, well, if you want them bad enough, you can go up there and get them, if you want them badly enough. Otherwise, you have to read the ones that are on reserve in the main library or in the department library. Remember then that you've got, essentially then you've got to know 6, quote, insects that affect the foliage or the fruit, foliage or fruit. We're going to cover altogether 3 problems with the roots. We covered phylloxera. We're covering nematodes today, and oak group fungus. That gives you 3 on the roots. And then we will have 3 leaf fungi, or fungus that affects the leaves. And then you're getting those 3 or 4 virus problems that you got in the lab yesterday and Monday and this afternoon. And I told you first, as an introduction, that Goheen would run this thing into the ground by giving you all the little minor viruses enough to confuse you. And I told you that the ones I want you to know are dead arm, and fan leaf, and what else? And leaf roll, of course. What am I dead arm? What am I doing? That was part of this morning's lecture. leaf roll, fan leaf. I had 4 of them I want you to know. What are the other two? Well, we used to give yellow mosaic, but remember this, and yellow vein. And the reason I used to call it 4 is that you've got to know this complex of vein banding and yellow mosaic, as it's called. And really, I think those are the only ones you really need to know. Of course, Goheen would argue strongly that you need to know corky bark, and he can show you out in the field how corky bark really knocks the indicator plant dead, but it's awful hard to recognize it out in the field. And Pierce's disease, which we have to put up here, thank you, which we have to put up here as a sort of a ditto line under it, because it now is no longer considered a virus. And those are the only ones I want you to know. In fact, I'm not real sure I worry about corky bark, but and I want you to know why each is important. Leaf roll is our most important virus because it is by far the most widespread, the most intensive in vineyards of the world. Lots of vineyards will run, well, I've seen a number of vineyards myself in which it is 100%. For example, Ruby Cabernet was released as a new variety in which it was completely infected with leaf roll. So every Ruby Cabernet vineyard that's 10 years old is 100% leaf roll. And Cardinal, which is, of course, one of our important table varieties, about 1/2 of its early release was budded onto old vines that had leaf roll. So you get mixtures of leaf roll in Cardinal, which is where it's really serious because it affects the color. And as Goheen did not, I don't think he indicated, neither did Ali, some E, that the original author on leaf roll estimated that in many vineyards of Germany, 80% of the vines had leaf roll. So leaf roll cuts the sugar and cuts the crop, and it is the most important virus in the world because it's the most widespread. But widespread is not the word I want. It's the most intensive also, and the most completely in vineyards. Fan leaf is an important virus. It does cut crop, but it isn't so widespread as leaf roll, and it isn't so, yeah, I guess it is just as bad on an individual vine, but it's far from being as widespread as leaf roll. And yellow vein, on the 2 or 3 varieties that it hits badly, which in particular are Carignan and Valdipinas, are the 2 in California. These 2 varieties are hit terrifically with yellow vein, and on individual vine, this is the worst virus. But fortunately, even in these 2 varieties in California, you may have an average of 1 or 2 vines per acre. Or maybe in some situations, I said an average of 1 or 2, it may go as high as 5 or 10 vines per acre. But where you have this virus, which is sometimes called fruitless Carignan, it's sometimes called fruitless Carignan. It means just that. You may get a pound of fruit. And a thing to keep in mind, especially in these areas around Lodi and Stockton, so forth, where we have Carignan and Valdipinas, is that the odd thing about this particular virus is that it knocks all the crop off. But it doesn't hurt the vigor of the vine. In fact, it's just that you'd use the pruning shears and took all the fruit off. So it grows terrifically vigorous. And you can see a block of Carignans along here where the vines are all that high, and all of a sudden you see one that big. And it's these big vines then that frequently have a yellow vein, as you find out in the vineyard in the lab the past days and this afternoon. Were you able to find any yellow vein? Dr. T, were you out yesterday? Those who were, were you able to see any yellow vein? It hadn't shown yet. It's a little early for it. And you only find about 1 or 2 leaves per vine with just a few specks on it. And we usually have to tell the class, don't pull those leaves off. We have 2 more labs coming, and there's only 1 or 2 in it. And so if that were a high rate of infection, or of yellow vein would be the most terrible virus that we could work with. But very fortunately, it is extremely rare. But when it does, sometimes people go out in the wintertime then to make cuttings, and these big vigorous vines make a lot of nice cuttings. So you can speed it from there. Well, this wasn't supposed to be a virus lecture because Dr. Alley's covering that, but I just thought that after listening to Monday afternoon's lecture that there were a few high points such as this, or I consider them high points, that I thought you ought to go over again. Getting back to this thing, I will put, for those of you really interested in looking at it, about 6 copies of this in our own department library. They can't keep us from doing that, at least till they find out about it. And we'll put that on a 2-hour reserve so that you might want to look through it. Okay, I said we had 3 soil problems, 3 fungus problems, and this group of virus problems that you're going to be held responsible for. So let's get into this matter then. We've covered phylloxera. Let's get into nematodes and see if we can cover that out this morning. And once again, as so often as I told you during this quarter, we're supposed to cover in 30 minutes what they offer 6 units on over in the nematology department. So where do we start? Well, we do know that nematodes are a big problem in the whole world. Well, first I might say, what are they? Some of you may not even know that. They're very small, worm-like organisms that live in the soil and in animals. And they vary from anything as small as from as small as 1/250th of an inch in length up to over 2 feet in some animal parasites. So there's hundreds and hundreds of types of nematodes that affect both all kinds of plants and animals. But what we want to narrow down to today is what do they do on grapes, because that's all we have time to talk about. And in California, we have 2 or 3 that are most important. The most important one of all, of course, is the root knot. The root knot nematode. And I might add to this general statement about the variation in size that generally the males are little needle-like organisms that are root knot, and the ones we're going to talk about this morning cannot be seen unless you have a microscope. So this is the male form, and it can usually move through the water, through the sand and so forth. And then we have the female that once it gets onto a root, this being a root, usually is more gourd-shaped or egg-shaped like this. And then it sticks its head into the root and feeds, and then exudes eggs, 500 to 1,000 eggs per female. And on root knot nematode, when this female begins to feed, it causes cancer-like growth of cells that finally embed the female. So the female doesn't move. The male does, but the female, once she starts to feed and lays eggs, the tissue grows out around her, and she stays there. So the root knot nematode, which is and unfortunately, you've got to remember this, Melodogeny incognita. Melodogeny, is that spelled right? No. Well, if I can't spell it, then you don't have to spell it. Melodogeny incognita is by far our most important parasitic nematode. As I say, perhaps fortunately, but unfortunately, the males can move freely, but the adult females, once they land, they stay where they are. So it's called a sedentary type of nematode. So the root knot is the most widespread so far as grapes are concerned. We have another one that's of some importance, but we don't really know how much damage it does, and that's the root lesion nematode. After sitting in the back of the room once, I find out that you can't see way back there unless you write it at the top of the board. The root lesion nematode, and this what do I want to say here now? What I want to say here is that the root lesion nematode is an endo-parasitic nematode. Endo-parasitic. An endo-parasitic, meaning that it moves inside the roots and then eats its way down through the cells, and as it eats out one cell, it moves on and so forth. Just a burrowing gopher through the root system. Now, why I hesitated was whether or not this was endo-parasitic. And it sort of puts you on the borderline, because you say, well, it attaches itself to the outside of the root. Therefore, you think it'd be ectoparasitic. But it is not, because with this tissue that grows out around it and encloses it, it essentially then becomes endoparasitic. So that's why I hesitate there trying to remember myself, because I always get confused on that. And then finally, and I won't even ask you to remember that this is Prerequisite and so forth, because I do want you to know this, though. Finally, we come to the third type, which is the Dagger Nematode. And you have to remember this, unfortunately, it's Xiphenema index. And the great importance of this nematode is that it is the vector, as you found out, I hope, in the lectures on virus. I had to leave before it was quite over. But it is the soil. It's a way that the fan leaf series here, I better put that up here, that this fan leaf it's the vector, or the way it is spread in the soil. The Dagger Nematode is a big nematode, and it's ectoparasitic, and it stabs its feeding tube deep enough so that it can get into the xylem, and it can transmit, it picks up this virus and can transmit it, just as I often say, mosquitoes transmit yellow fever. So it digs into the roots and then transmits it in this form. So one other thing about it, it's a big nematode, it's ectoparasitic, and it's sort of a in-between phylloxera and the root knot nematode, or even this root lesion nematode, because, as I say, it's bigger and it's definitely ectoparasitic, and it feeds deeper and acts quite a bit like phylloxera in its nature and the way in which we can kill it. That's one reason why we can kill it, as I'll talk about in a moment, with fumigation. So those are the three that you need to know about. And then the other thing about these Dagger nematodes, since it is definitely on the outside, it is migratory. That is, it can move through and does move through the soil. So we have those three general types that affect grapes. Okay, as I say, they're very small. If you want to put it down, the adult male is said to be about 0.15 millimeters in length. And the female of the root knot nematode, this one that digs into here, is big enough so that under good light and with good eyes, you can see them. Yes, right. No, this, good point. This is the root knot nematode, 0.15 millimeters in length. It's the root knot. And just like we were talking about with the phylloxera, they can build up rather rapidly. Of course, it depends on the soil temperature, but an egg, from egg to female, yes. Well, we're talking mainly about root knot in this particular case, right? That you can have 5 to 10 generations a year, and as I say, each female can lay up 500 to 1,000 eggs. And they do not have to mate. The female, like the phylloxera, can lay the eggs and their fertile eggs and so on without mating. So on an average, for the root knot nematode, at our average soil temperature, it takes about 20 days for a complete cycle to go from a mother to through the egg larva and to egg-laying mother again. And one of the bad things about nematodes, which we do not have in phylloxera, is that many of them form what we call a cyst. I mentioned this last time, a cyst stage. And the cyst is a sort of a, told you, sort of a hard shell about it, and they can go then for several years without water. It's a sort of a nature's way of preserving the species. And this is one of the reasons why it's so extremely difficult to completely wipe out nematodes. This is the root knot, again, that we're talking about mainly, unless I mention otherwise, and I'm mentioning otherwise now, in that fortunately the dagger nematode does not form a cyst. This dagger nematode, Zephyrema index, does not form the cyst stage. So our fumigation and our controls for it have to be a little bit, see, I say it's sort of an in-between phylloxera and the root knot nematode in many of its characteristics. How to hit the high points here is what I'm trying to get at here. Now, before I forget it, I want to interrupt right here. I'm teaching from my old notes plus a bulletin which is just in the last 2 weeks come out on nematodes and the control in vineyards. So this bulletin is circular 533, and all you got to say is nematodes. And it should say revised, yes. And that's the main point you want to get, is be sure you get the revised copy, and that's 1973. So circular 533, nematodes revised 1973 by Rasky and Hart and Casamatas, but you don't need to know the authors. You can get this again out of our extension office upstairs or over in Marac Hall. And this gives the whole complete story, in which I'm just trying to pick out, and shows you pictures of the nematodes and tells you their action and gives you the various types. Shows you the damage, which we're going to look at in a moment, and also gives you all the various and sundry treatments, including the rootstocks which can be used. So it's a pretty good bulletin.
Back on the root lesion type, does that also form a cyst, or does that move freely through the soil?
Well, whether they move freely through the soil doesn't have anything to do with whether it makes a cyst or not. But so far as I know, yeah. Oh. Well, I think the lesion would form cyst, but I'm not sure. I'm not sure on it at all. Okay, all right, we get around, let's look at the damage. That's the best thing to do, start, and then we worry about the controls. You've got the 3 types that you got to know about, and you got to know these 2 scientific names. You know the root and whether or not they're ecto or endoparasitic. Okay. Let's see some of the damage they do. And remember that, this is the other point I want to make about this dagger nematode. When I say it has a lot of the characteristics of phylloxera, is that it thrives in heavy soil, just as phylloxera does. And that's why it's a problem in the coastal counties, where they have heavy soils. So in many ways, it's sort of an in-between those 2. Okay, let's see a few slides here and see what we got. I said that nematodes generally, and this is root knot now, we're not talking about dagger nematodes. Incidentally, just as a personal remark and so forth, it took me a long time to believe that we could have any sort of a nematode problem in Napa Valley. But Rasky's work over the years has finally proven off that he's absolutely right, because I couldn't imagine nematodes in that 80% clay, bale clay soils. But anyway, normally our nematodes, especially our root knot, are in sandy soils. And here's a shot from Coachella Valley where it's about 90% sand, showing you a nematode spot where the vines have really been wiped out with nematode action on the roots. Next slide. And here's what it looked like on the roots. This is a rooting of a nursery. I was telling you the other day that you have to, if you make a mistake and plant your nursery in a situation like this where you get these root knot nematodes, these knots along here, we'll see a better close-up in a moment, but you see these big swellings and so on are just a constant overlapping series of females sitting side by side, making a continuous enlarged root. Sometimes you'll just see beads like this, and inside those beads, that female is sitting there with the cancer cells causing the growth around her and enclosing her as she lays her eggs. Okay, next slide. And this is the way the roots look with root knot nematode as a close-up. Now notice, you ought to be able to tell the difference between phylloxera roots and nematode roots. What's the difference? You don't have those goosenecks. You don't have those goosenecks like this out at the end of the little roots. You just have the big knots. But if you get those little, get very many of those little crooks there, it might be one right there. But one more similarity, which you might throw into your notes with the Dagger Nematode, is it makes that type of gooseneck that the phylloxera does. So why didn't it join the phylloxera tribe and be done with it? But anyway, here then gives you an idea of the root knot nematode. Here's some of the more extreme types that you can often see on the roots. Now you ought to look at that a little bit, because so often when we go out and talk to farmers and ask them if they got nematodes, they say don't have any idea what one looks like. And you dig up a few roots and they're just amazed to see what what nematodes do to cause these root knots. See, they can be pretty big. They can go 4 or 5 times diameter. Okay, next slide. Now, we're going to talk about controls in a moment, but we'll get these slides out of the way. And of course, the best control of all for nematodes is to use, unless we're worrying about Dagger, which we'll discuss a little specifically in a moment, but is to use rootstocks, which are resistant. And last time, some of you asked me about whether or not some of these rootstocks for phylloxera were absolutely immune or just tolerant. And I don't know what I was thinking about. Of course, it's a varying degree of tolerance. Otherwise, why would St. George be more resistant than AXR, and AXR a little bit more resistant than 1613, and so forth? It's just that they have varying degrees of tolerance. But in this example here, you can see this is almost immune. These are Dr. Leiter's rootings grown in the greenhouse in tanks where he just saturates the soil with tomato plants infected with these nematodes, and then plants his seedlings from his breeding projects to see which ones will pick up the root knot nematodes and which ones will be tolerant or resistant or immune, as this seems to be. So in this particular cross, he got a very good possibility for a rootstock, provided everything else goes along, grafting and rooting of cuttings and graft takes and compatibility and so on. But you can see over here one where we have Vitis vinifera. You see rupestris is St. George, and you take vinifera and rupestris, and I told you St. George had no resistance to nematodes, and neither does vinifera. And boy, when you cross those two, you can really get some susceptibility to nematodes. So the best control is rootstocks for root knot and lesion nematode and so on. But let's see the next slide then. And this is a shot taken down at Delano of rootstock trial of Dr. Leiter's, in which this is Dog Ridge rootstock, which is very resistant to nematodes and quite vigorous, and here's own roots. And you see the difference then caused in just a moderate situation with nematodes. So I think you got one more in that series or not? Oh, 2 more. Okay, now This is the equipment we use. I'll give this so that we can talk about it later, but this is equipment for fumigating the soil, for nematode control, and this is a shot taken over in Napa Valley where they're fumigating to control Dagger Nematode. I'll give you all that dope in a moment, but I wanted to show you pictures of the equipment. It's an expensive operation. It costs about $400 an acre to control it, and you see the type of equipment. It takes a lot of material to control it. I'll tell you in a moment again, but right now I'll tell you that the general control, the most popular control, is carbon bisulfide, CS2, at about 300 gallons per acre. And each gallon weighs over 10 pounds, so you're putting 1.5 tons of carbon bisulfide to the acre. 1.5 tons, so you see why you have such big equipment to do it. One more slide then. And this is just another shot showing the type of equipment you have to use. You have to chisel first and chisel deep, loosen up the soil, and then apply these fantastically high rates of material to control Dagger Nemat, or to wipe it out for planting. And that's all the slides in that, okay? Okay, now we'll essentially start over here on one little part of it. Yeah, please. Okay, let's say the best control is to use rootstocks, but you've got this problem. If you've got old vineyard land in Napa Valley, often over in Napa Valley we have a fairly high infestation rate or infected rate of fan leaf virus, which I tell you is fairly serious in this situation. And if you're paying $3,000 an acre for the land, an old vineyard site, you don't want to go in and replant without being sure that you wiped out all possibility of getting fan leaf from Dagger Nematode. In addition to that, which we're going to try to get time to talk about, probably won't, many of those old vineyards have oak root fungus in them. So that you want to get rid of both of these things. And then the additional point is that that if you want to use methyl bromide, you can wipe out all weeds, seeds, morning glory seeds, what have you, and so forth by fumigating with methyl bromide. So you have your choice then of fumigating with carbon bisulfide, that's CS2, or with ethyl, what is it I want? methyl, I'm looking at something else here and lost, with methyl bromide. I don't know why I have something else written here. With methyl bromide, that's CH3Br. Now, both of these materials, while methyl bromide has just come into the picture in the past 5 years or so, and there's still some experimental work going on to see how it works out. And we're not sure yet which is going to be better for controlling Dagger Nematode, whether it's methyl bromide or CS2. But the feeling here at this campus, because it's, I guess because Dr. Rasky is partial to it, is to use CS2 for treating for Dagger Nematode. And, well, the point is this, that you can use CS2 and wipe out oak root fungus at the same time. But these other materials, which are most commonly used where you just have no possibility or no likelihood of oak root fungus, is to use what we call 1,3-D. I mean, that's the easiest way to remember it, 1,3-D. But this is often called DD, capital D, hyphen D, which is one company's trademark.
Will methyl bromide kill oak root fungus?
That's the main reason. That's one of the reasons why it's coming in. It's a new treatment, but we still got problems. All right, DD, or Telone, T-E-L-O-N-E, which is another company's trademark, or one called VEN116b, which is another company's trademark, all for materials. So it's DD, VEN116b, and Telone. All are trademark names for a material which essentially the active ingredient is called 1,3-D. And if some of you are going to hold up your hand and want to know what 1,3-D is, it's 1,3-dichloropropene. So, and it's a mixture of dichloropropene and dichloropropane. Now, the standard treatment then for using this material, let's go first. I'm getting you confused now, but let's cover this while we're at it. The way to treat with this is to have soil moisture at about 2/3 field capacity. 2/3, 3/4 field capacity. And this is Dr. Raske's favorite treatment for Dagger Nematode, is to give it 200 gallons at 30 inches, and then to cross that with 50 gallons at about 8 to 12 inches. And this is supposed to be done, as I say, fairly when the soil moisture is about 60% of field capacity.
Is there any reason why you can't apply those both at the same time?
You mean, well, you do, well, if you got the equipment, but you'd have to take some monkey business equipment to have one set going at 30 inches and one at 10. But I guess you could design the equipment that way. But the equipment isn't set up that way. So they do it one way with 30 inches and go back the other way at 10 inches, or 10 or 12 inches. Now that is if you want to use this 1,3-D. Now if you want to use methylbromide, it takes about 400 pounds of methylbromide injected to about 24 inches. And in this case, you've got to have the soil as dry as possible. So you've got to do it real late in the fall. You have to sort of guess when you're going to get the soil as dry as possible to get down. Some people plant a cover crop of safflower or something like this to dry the soil out to make sure that it's as dry as possible so the methylbromide can permeate through this dry soil. And of course, that's the one that requires the tarp that you saw out in one of the labs. You've got to cover it with plastic. It can be usually pretty thin, just 1 or 2 mils thick. We'll hold it, and this should be on it for about a week in order to make sure that you get good downward permeability and no loss to the atmosphere. Okay, the third material is carbon bisulfide, which I said that was the old standard control. And this is mainly used in the coastal counties, remember, for dagger nematode and for oak root fungus. And where you have possibilities of oak root fungus, either methylbromide or carbon bisulfide ought to be used. As I said in the picture there, when we're showing the slides, it takes about 300 gallons to the acre, which is 1.5 tons. And this doesn't have to go very deep. If the soil is worked up pretty well, you can just inject this down to about 6 or 8 inches and take care of it. But you've got to have the soil, again, as dry as, in this case, as dry as possible. Remember, with methylbromide and carbon bisulfide, you've got to have very dry soil. With these materials, you want to have normal soil moisture. Don't ask me why that's the situation, but that's what I'm told. Now, one of the odd little things about carbon bisulfide is that you can hardly use it in rocky soil. Otherwise, you blow up the place. I thought they'd call somebody to look up. Because if you've got rocky soil and it's dry, and those injectors, you saw those big prongs sticking down to root down 10 inches or so to apply it, it's explosive. And if those injector tubes hit rocks and cause sparks underground, you've got a whole bunch of little landmine explosions going on behind you. Not bad, but it's something you have to keep in mind. The carbon bisulfide is explosive. So you can't use it, it's not wise to use it on real rocky soil because of this problem. So if you buy an old vineyard land, you've got this problem then in the coastal counties of dagger nematode and the possibility of oak root fungus, in which you've got to use then either methylbromide or carbon bisulfide. If you're just over in the coastal counties and want to wipe out nematodes, you could use any of these three. So it's these two for the coastal, it's CS2 or methylbromide for the coastal counties, and 1,3-D for the sandy soils of the interior valley, just to wipe out root knot as well as possible. Okay, and then the question always comes up, and I mentioned it briefly when we were talking about soils, in which we get the phone calls quite often from people who are buying land and say, well, I don't own an old grape vineyard. I bought an old prune orchard, or I've got virgin land in the coastal counties, which may or may not have oak root fungus. Should I go ahead and spend $400 an acre to fumigate the whole 40 acres? And remember, I went over this with you in class, and it was my argument that unless you've had it in grapes, old grapes, that you're better off just to go ahead and plant it and then look for the oak root fungus spots later, because you don't have, see, Dagger Nematode, even if it's present in a prune orchard, it isn't going to hurt you because it doesn't have fan leaf virus from grapes, because it has to pick up the fan leaf from a diseased grapevine. So you don't worry about whether there's any Dagger nematodes there or not. If you're going to go in and plant virus-free vines, which you'd better do if you're going to spend that much money for land today, so you don't have to worry about it on anything except old grape land. And as far as oak root fungus is concerned, if you're buying an old prune orchard and you see dead spots because oak root fungus affects a lot of plants, then you might want to fumigate, depending on what percentage of that 40 acres you bought looks like it has dead areas in it. But if it hasn't been planted anything, then my suggestion is go ahead and plant it. Don't worry about the fumigation. And then you can always go back in these small areas. The big argument that's been given to me is that, well, if I get an acre later on, I can't get a contractor to come in and do just 1 acre or a half an acre. But if you get oak root fungus spots, usually about the size of this room when they start out, and you can take care of oak root fungus with carbon bisulfide, I'll discuss oak root fungus in a moment, by putting it on by hand, because they have little hand injectors that you can go along like some of these things that you put in to aerate your lawn, and inject the carbon bisulfide on 18-inch centers or so forth, about 1 ounce per shot, and wipe it out by hand if you have to, rather than spend $400 an acre over your 40 acres just to be sure that you've covered it.
I heard that they had a hard time eradicating oak root fungus because it can be in any kind of root.
Yeah, well, we'll discuss oak root fungus in just one moment. I just want to cover it while we're talking about this material. Yeah.
I have a question about the Dagger Nemat. So will they transmit the family complex to vines with resistant rootstock?
Yeah, yeah. Well, there's no resistant rootstock to viruses that we know.
Yeah, but I mean the nematodes they can inject.
Oh, I see what you mean. Yeah, yeah. But I don't know. Unfortunately, the rootstock seed that we use in the coastal counties are not nematode resistant. They're phylloxera resistant. But you got a good point. What would you do if you took dog ridge over there, and whether or not Dagger nematode will affect dog ridge? I don't know. That's one I've got to. I learn something every lecture.
Nematodes come in irrigation water?
Right. See, they can move easy in irrigation water. That's what I said. They're migratory. At least they may. Well, you don't. You put on rootstocks. But on this Dagger nematode, of course, you're not getting. There you're using your own water, well water, or water from your own reservoir. So suppose if you fumigate and so forth, you should have taken care of that. In other words, the water isn't.
Is there any way of analyzing the water to determine whether the nematodes in it, if there are any, are some really harmful?
Yeah, you can send in samples to the nematology department, and they have a service through the farm advisor and so on to see if the soil or the water has nematodes in it. Norman Ferrari, who works for Dr. Leiter, does a lot of analyzing of soils himself. And that's where you can go in and see these things wiggling around like snakes under the mic. All right, now, as I say, the real control then for nematodes is to use rootstocks, and we've mentioned these before. The two most resistant ones of all are Dog Ridge and Salt Creek. Remember what I've also said about these? Is the Dog Ridge is the most vigorous, and therefore it's so vigorous that it should usually be used only on what Dr. Alley likes to refer to as pure sand. Otherwise, you have so much vigor that you have problems controlling it and also getting fruit. Then you can come down to Salt Creek. It's the other one that is a long known rootstock, and it is not quite so vigorous as Dogwood. So if you had a place where you had just a little bit of soil, you might want to use Salt Creek. And then we get down to this one I mentioned, that we mentioned. I'm taking them down now more or less in order of vigor and fruiting, or harmony, which we mentioned last time, having some resistance to both phylloxera and nematodes. And below that we have 1613. So those are the main rootstocks that you need to worry about for nematode resistance. And this new one, which has just come out, and we don't know much about it, but supposedly it's an improvement over harmony, the one called freedom. As I said, I'm not going to require you to know the parents and so on of these. Maybe I should on this one here. These are both Vitis champagne. So at least remember one, and you know that 1613 is a hybrid between a fruiting variety, Solanus boethello. So Solanus is the native species of Othello, so you don't need to know that except to know that it is a hybrid. And to remember that St. George and AXR have no resistance to nematodes. And that's about all I'm going to ask you to remember on those. Okay? Any more questions then on nematodes? Because we're out of time. See, I was going to cover another subject. Well, next time then we'll pick up oak root fungus, which will be the third root organism, third and last root organism that we'll cover. And then we'll try to cover the 3 fungus problems next Wednesday. Remember, Monday's a holiday.